Hello, and welcome to our educational lecture series at the Center for Psychedelic Psychotherapy and Trauma Research. Today, I have the pleasure of introducing Gül Dölen. Dr. Dölen is an associate professor of neuroscience at Johns Hopkins. She earned her MD PhD at Brown University and MIT, studying the pathogenesis of autism. She then completed her postdoc at Stanford, where she conducted seminal work on the neural circuits of social reward learning. The focus of Dr. Dölen's lab at Hopkins is to understand how plasticity and neuromodulation govern social behavior in health and disease. She is a leader in illustrating how MDMA may induce pro-social behaviors across species. Recently, her lab has made the groundbreaking discovery that MDMA can reopen a novel critical period of brain plasticity in mice. Building on this discovery, Dr. Dölen has initiated the Fathom project, which tests the hypothesis that psychedelic drugs as a class act to reopen multiple critical periods across the brain. Dr. Dölen also serves as on the executive advisory board for the International Arts and Minds Lab, a translational research approach designed to solve intractable problems in health, well-being, and learning through the arts. Her passionate curiosity to understand the mind from philosophical, spiritual, linguistic, and artistic perspectives shines through her work. At the end of her lecture, we'll have time for questions. So please type your questions in the Q&A box, and we'll ask you if you want to come on screen to ask. Without further ado, Dr. Dölen.
All right, thank you so much for the invitation, and I'm really glad to be here today. I'm sorry that we're not in person, but actually having this opportunity to do it virtually actually, I think, made this possible today. So I'm really grateful to have virtual after all. So without further ado, let me just, so today I am going to tell you guys about some new work that we have just submitted that looks at critical period reopening as a sort of universal mechanism that generalizes across psychedelics, and give you some hints about where we're heading in terms of extending that insight to other critical periods for some novel therapeutic opportunities based on those mechanisms. So my lab, as Lauren mentioned, in general, we work on social behaviors. We look at it from a number of different perspectives. The main thrusts of the lab are trying to understand critical periods, looking at social brain evolution, and then also understanding this sort of neural circuitry of different subtypes of social behavior. And the evolution and the parallel processing studies are really exciting, but today I'm going to talk about our work on critical periods. So, for those of you who are not familiar with critical periods or sensitive periods, as they're sometimes called, there are a ton of them. They were first described by Konrad Lorenz, describing this imprinting behavior in snow geese. And what he observed is that snow geese will form a lasting attachment to their moms, or if their moms are not around, if there's another moving object in the vicinity or another person, in this case Konrad Lorenz himself, then they will form that lasting attachment, but only if they are exposed to that stimulus, mom, Konrad Lorenz flying plane, if they're exposed during the first 24 to 48 hours after hatching. After that, that window of time is sort of closed, and those stimuli are no longer rewarding or causing that long-term learned attachment. And so he called that window of time the critical period. And since then, we have discovered many more critical periods. I think most people will be familiar with the critical period for language learning, but there's also critical periods for song learning in birds, sensory reorganization in mice. So this is somatosensory, but also visual system reorganization. And then from a clinical perspective, there is evidence that there are critical periods for reorganizing the brain after brain injury. So for example, stroke in this case is what this paper is looking at. And so these critical periods, people have known about them. Most of what we know about them, actually about the mechanisms underlying critical periods, comes from, pioneering studies by Hubel and Wiesel, looking at the ocular dominance plasticity critical period. And I'm not going to go into too much detail about this. I just want to kind of orient you to a couple of features, which is that in this case, the plasticity that's being induced is following a monocular deprivation. And then before they monocularly deprive, they record from the visual cortex and get a proportion of responses from each of the two eyes. And then after, typically 2 to 3 days of monocular deprivation, they record again and figure out how much reorganization has happened in the visual cortex following that manipulation. And when they do that, they get a lot of changes, a lot of reorganization of the cortex in early development that peaks sort of around adolescence. And then that ability to reorganize the cortex from that manipulation declines. And by adulthood, that critical period is closed. And what people have noticed is that through decades of study on this mechanism is that this developmental regulation of the ability to induce this kind of plastic change in the brain seems to be correlated to, downregulation of some things, upregulation of other things. And in general, I think the three main hypotheses that people have really focused on are metaplasticity, which is not exactly the same as hyperplasticity, and I'll get back to that later, but the ability to induce plasticity seems to be the thing that's changing. And that's been correlated to changes in receptor expression and downstream of that, biochemical changes. Other people have focused one step further and said, well, changes across development in inhibitory, excitatory balance, the maturation of inhibition as the animals get older is part of what enables this critical period to be constrained to this developmental window. And then what seems to be sort of the last step is this regulation of the extracellular matrix by things like the metallomatrix proteases, which seem to go down across development, and then sort of the upregulation of, most people have focused in the visual cortex on the perineuronal net, which is a specialization of the extracellular matrix specific to PV cells. So that's kind of the general sense of what people have gotten about mechanism. And the reason that neuroscientists, and especially translational neuroscientists, have been so interested in this understanding the mechanisms underlying these critical periods is that I think that probably since the very discovery of this concept of critical periods, there has been this intuition that the reason that we keep failing in clinical trials, even when we think we've got a handle on the mechanism of a disease, is that by the time we intervene, the relevant critical period for the clinical trial we're trying to do has already closed. And so if there was some way to reopen those critical periods, then we might have a better shot at being able to correct whatever imbalance that we are focused on. So in the visual system, correcting by removing cataracts, and then reopening the critical period. So under those corrected circumstances, the brain can adapt and learn the proper visual environment. And so this has been sort of the goal, and there have been a number of different approaches people have taken. I'm not going to go into too much detail on that. Instead, what I'm going to do is tell you about a new critical period that my lab discovered. So this critical period is the critical period for social reward learning. And, it looks sort of similar. This is the pooled data across something like over 500 male mice. We have a very similar regression curve for fitted to the female data, which we have, slightly less, like 450 animals. And this is across 15 different ages across development. And because this is mice, we can do this kind of extensive study to really characterize the critical period. And the way we measure this is it's a behavioral critical period. So we're using a old assay that's been around for 20, 30 years. And that was more recently adapted to be looking at the reward value or reward learning from social interactions, not by us, by this group that published this paper in 2007. And basically the idea is that what we're testing is the learned association between a bedding that they've never seen before and either the social condition or the isolate condition. And if in the post-conditioning trial, they spend significantly more time in the bedding that they learn to associate with the social condition, then we take that as evidence that they have a preference for that stimulus, and that and that they were able to learn that association between the social environment and this novel bedding. And we've done, a ton of controls around this to really convince ourselves that this is a critical period. But the other part of why we believe that this is sort of an honest to goodness critical period is that we were able to identify a metaplastic mechanism that seems to correspond to the behavioral plasticity, the change in the ability to learn correlated to metaplasticity across development. And so when I was a postdoc, I had discovered a new form of oxytocin-mediated synaptic plasticity called long-term depression, OT-LTD, long-term depression in the nucleus accumbens. And what Roman, who was the postdoc, who was the lead on this project, showed is that oxytocin-mediated LTD is robust in the juvenile animals. So at postnatal day 40, you bath apply oxytocin for 10 minutes and then you wash it out and you get this robust LTD. But if you do the same manipulation in slices cut from adult animals, then you don't get that LTD. And so that developmental change in the ability to induce oxytocin LTD is metaplasticity, or downregulation of oxytocinergic synaptic plasticity. And it's interesting because even though in our previous work we had linked oxytocin-mediated plasticity to serotonin 1B plasticity, we saw no evidence that serotonin 1B plasticity is developmentally downregulated. And so even though they're linked, they're mechanistically also separable. And this has implications for interpreting our data as well, which I'm happy to talk about if anybody has questions. Okay, so having discovered this sort of metaplastic correlate to our critical period, we were of course interested in trying to reopen the critical period. And we knew from a variety of sources, both, sort of the literature surrounding recreational use, some work from Alexander and Ann Shulgin using these drugs, using MDMA for therapy, that the empathogen 3,4-methylene deoxymethamphetamine, or MDMA, ecstasy, E, X, Molly, these are all synonyms for this drug that has these very robust pro-social effects. And, we knew about this and we thought, well, wouldn't it be great if we could use MDMA to reopen our critical period? So we tested this, and we took a different approach than most of the other people at the time who were focusing on trying to understand the pro-social effects of MDMA, and also different from the approach that we took in our studies of the octopus. What we did here is we gave the MDMA and then we waited for 48 hours. So we wanted to wait until all of the acute subjective effects of the MDMA had worn out and just test the sort of long-term consequences of this very robust manipulation. And what we found is that if we, in that 48-hour time point, we see this robust return of the ability to learn from the social environment. So we interpret this as reopening of this social reward learning critical period with MDMA, but not saline. And that was super exciting to us. And we wanted to know whether or not this manipulation, the same pre-treatment 48 hours earlier with MDMA, also impacted our metaplasticity mechanism that we were linking to this behavioral plasticity. And so what we showed is that in the animals that were pre-treated with MDMA, when we tried to induce oxytocin-mediated LTD, we were able, once again, and remember these are the adult mice, so in the adult mice, oxytocin plasticity is gone, but in the ones that had been pre-treated with MDMA, this oxytocin LTD has been restored. The ability to induce plasticity, oxytocin plasticity had been restored. And I just want to take a second here to remind you that this is not the same thing as hyperplasticity, right? So hyperplasticity is what we see with cocaine, right? So when you give cocaine, and especially in the nucleus accumbens, but also in the medial prefrontal cortex, which is part of the mesocortical limbic reward circuit, cocaine induces massive hyperplasticity, which you can see in the sort of mini EPSC recordings. You see the big change in the frequency, but not the amplitude. And you can also see it at the level of morphology. So you get changes in dendritic arborization and the amount of dendritic spines. And this hyperplasticity is thought to be the basis of cocaine's addictive properties, okay? And so it was really important to us to be able to differentiate, but if we're gonna make the claim that the MDMA-induced reopening of the critical period has something to do with its therapeutic effects, we want to be able to differentiate it from cocaine, which does not share the therapeutic profile of MDMA for treating PTSD. And so we looked to see whether or not cocaine is able to reopen our social reward learning critical period, and it doesn't. Okay, so this is a really important negative control. It's probably, I think, the most important control that we've done. We've looked at it at a number of different doses, a number of different time points, and basically we've convinced ourselves that cocaine does not reopen the social reward learning critical period. So in this is part of our evidence that we can sort of mechanistically differentiate between plasticity and critical period reopening as two different concepts, and they are governed by different mechanisms. So I'm going to show you in a second some more evidence about the metaplasticity. So, as we were looking at this, we wanted to know, can we recapitulate other parts of what people have noticed in the human clinical trials using MDMA to treat PTSD. So this is just a redrawing of some of the data presented by the Mithoffers, showing the really robust, rapid effects of MDMA that seem to last for months to years out after the acute subjective effects of MDMA have worn out. So we went ahead and looked at the acute subjective effect, the ability of the time course of the ability of MDMA to reopen. And we also noticed that it was very durable. And I can talk a little bit more about this at the end when I have all of the data together. So then we also wanted to test this other aspect of it. And I think this is probably the part of the story that I'm most interested in moving forward, is this idea that, psychedelics are not just next generation SSRIs. They're not just next generation anxiolytics. What makes them special is that they seem to be able to induce like a cure that doesn't, you can't just give a person a pill and expect them to be cured. In fact, the way that people are typically using ketamine these days is, they're not paying too much attention to the set and setting or the context dependence of it. But the MDMA assisted psychotherapy for PTSD, those trials really emphasize the therapeutic context. And we've known this about psychedelics for a while, that the context really matters. And, the psychonauts call it set and setting, the neuroscientists call it context. And we wanted to know whether or not that context dependence is something that we can recapitulate in the critical period reopening. And so just like the therapeutic effects are context dependent, so you can't go to a rave and expect that your PTSD is just going to be cured. You actually have to be focused on the trauma and be working through those traumatic memories. Similarly, if we give MDMA and not in a social context, so in isolation, then we don't reopen the social critical period. And we think this has implications for understanding the mechanism, but also for how we're going to apply this therapeutically. Okay, so this was sort of the Nature paper from a couple of years ago. And moving forward, we wanted to know, our original thinking about all of this was that the reason that MDMA is reopening the social critical period is because it has these really robust acute prosocial effects, and that's somehow triggering the social circuitry to reset itself. But what we also noticed is that, MDMA, even though it's different from my other psychedelics because it has this prosocial property, this empathogenic quality, and other psychedelics like ketamine or dissociatives, or, psychedelic can be classified as hallucinogenic or onirogenic. The thing that all of these psychedelics share is that they induce what's called an altered state of consciousness. So our idea is that if the ability of MDMA to reopen the critical period was because of the prosocial properties, then only MDMA and none of these other psychedelics that don't have acute prosocial qualities should be able to reopen. If, on the other hand, the reason the MDMA is able to reopen critical periods is just the fact that this altered state of consciousness that all of the psychedelics share is just what it feels like to reopen critical periods, then you might expect that all of the psychedelics would be able to reopen our critical period, and that we might be able to extend to other critical periods that have nothing to do with social, okay? And so we started to look at that, and we were really surprised, honestly, that basically all of the psychedelics reopen. So psilocybin, which is not acutely prosocial, nobody's doing cuddle puddles on psilocybin, or LSD or ketamine, right? And yet psilocybin was able to reopen this critical period. LSD was able to reopen this critical period. Both ketamine and ibogaine were able to reopen the critical period. Okay, so they all do it, and this is at the 48-hour time point right after. But we also knew from the literature that there's a huge amount of variability across psychedelics, both in terms of their therapeutic time course and in terms of the time course of the acute subjective effects. So this is just my summary of the human literature around the acute subjective effects of psychedelics. And under here is my sort of color-coded reference guide if anybody wants to look up these papers. I think some of these papers could, it would be nice to have some more robust data on these and some repeats. I think that would be a really great thing for people to make some more papers on this, because some of the times the data is pretty sparse. We're talking about 10 patients and some anecdotal reports, but still, basically what we know is that there's a huge amount of variability. So ketamine the acute subjective effects last about 30 minutes to 2 hours. Psilocybin and MDMA are 3 to 6 hours. LSD is 8 to 10 hours. And ibogaine is really sort of this amazing, extremely long trip, 36 to 72 hours. And there's some correlation between, how long the trip lasts and how durable the therapeutic effects, with ketamine's effects lasting about a week, and people reporting 3 to 4 years out for MDMA, LSD, and ibogaine. Okay, so we wanted to know, how does that relate to our critical period, if there's some correlation between that. And so what we did is we just looked at 1, 2, 3, and 4 weeks after we gave the psychedelic and just measured whether or not the critical period was still reopened. And so ketamine, the critical period at 1 week is closed, but psilocybin, it's still open. At 2 weeks, LSD and psilocybin are still open. At 3 weeks, LSD is open, but psilocybin is closed again. And at 4 weeks, LSD is closed, but ketamine is still open. And so when we sort of summarize all of that data in the critical period open state now, what we see is this remarkable sort of proportional response between the ability of these psychedelics to reopen the duration of the open state and the duration of the acute subjective effects. And so this is probably, like, if there's one take-home message slide, this is it, because especially for a sort of clinically oriented audience, because what this is telling us is a couple things. One, it lends further support to this idea that what it feels like to reopen critical periods is just what it feels like to be in that altered state of consciousness, right? Second, it tells us that somehow the proportionality is telling us that somehow these are mechanistically related to each other. How long you stay in that open state is dependent on how long the trip was. And so all of these companies that are being developed to sort of shorten the duration of the critical period might be turning a rock star drug like ibogaine, which is able to cure heroin addiction, and turning it into something much less effective, right? So that would be I would raise a concern that we don't want to overdo it on trying to get rid of the psychedelic side effects, because they seem to be related to each other, the ability to reopen this critical period. And then the third thing I would say is that this should be a reminder to anybody who's doing these clinical trials that, once the patient goes home after the psychedelic has worn off, they're actually still very much in that vulnerable state, right? And they are still very much, susceptible to the world and suggestible and vulnerable and in that malleable state of that open state of that critical period for weeks, right? I mean, translating mouse time to human times is notoriously difficult, especially when you're talking about developmental things. But for MDMA, for example, it's at least 2 weeks out and probably more like 2 months out from the big MDMA experience. Would be our prediction based on these mice, and so on these studies. And so I think that as we move forward, we're probably going to need to spend a little bit more time making sure that we are not sort of giving people these drugs and then not taking care of them in the post acute subjective effects lingering open state of the critical period. And I think that lingering open state is actually where a huge amount of the therapeutic work is also happening. Okay, so then I just want to return now to this concept of metaplasticity. So, I showed you that we had evidence that MDMA is inducing metaplasticity, and we wanted to check whether or not we were seeing the same thing for all the other psychedelics and whether or not we were seeing the same thing for cocaine. And so I'm not going to go through a huge amount of detail here, but we knew that it should be presynaptic. So we were expecting changes in frequency and not amplitude if oxytocin plasticity has come back. So we inject the animal with, whichever drug we're going to look at, and then we wait for 48 hours, and then we cut brain slices, and we allow them to recover, and then we do that 10-minute bath application to induce oxytocin plasticity, and then we record these minis. And what we saw is that oxytocin plasticity that we see in juveniles but not in adults, we don't see after pre-injection with either saline or cocaine, but we do see it with MDMA, so we replicated our previous findings, and we also see it with LSD, ketamine, and ibogaine. Okay, so this is all further evidence that the psychedelics are inducing metaplasticity, and suggests that sort of the social oxytocin part of it is just sort of a red herring. It's just any metaplasticity will do. And then, you've probably heard of, there are some claims out there that, these drugs are plastogenics or psychoplastogens, but I sort of hate that term. I hate psychoplastogen. I think it's a misunderstanding of what, well, first of all, where the terms come from, because cocaine is a psychoactive plastogen, right? So psychoplastogen could equally, apply to cocaine. That doesn't have, so it's not a term that accurately distinguishes cocaine from the psychedelics. Second of all, that claim about the plastinogenic effects were based on studies done in vitro mostly. So, dissociated neurons in a dish, and those dissociated neurons in a dish don't have extracellular matrix. They don't have the maturation of inhibition. So none of the mechanisms that we think are relevant for critical period closing, are recapitulated in a dish. Then there was one sort of ex vivo study that they did, which we were not able to reproduce. And so this is just evidence that there's no hyperplasticity. So if you don't give bath application of oxytocin, we don't see any evidence that there's hyperplasticity. When we have taken a look at some of the other papers that I think were much more carefully done, like from Connor Liston and from Alex Kwan and Lisa Montegia, there, I think what they're the much more subtle plasticity phenotypes that they're reporting, I actually think are what you would expect if you were inducing metaplasticity. So if you look carefully at those papers, they're not showing like sprouting the way that we see with cocaine. They're showing if there was a synapse here and then you induced, and then you gave a stressful event and they went away, and then you give psychedelics, they come back, right? So they're not inducing hyperplastic changes. It looks more like what you would expect based on a metaplastic mechanism. So I think that that's going to be really important moving forward, especially when we start thinking about the relevance of the context dependence. And as we start developing new drugs that we want to say have a better therapeutic profile. And we also want to avoid turning a drug that is not addictive into a drug that is addictive, because if we use plasticity or hyperplasticity as the end point that we're going after, we could potentially be turning these drugs into addictive drugs, you know, sort of because we used the wrong biomarker for the behavioral, you know, the therapeutic consequences. So that's my sort of soapbox spiel on, I hate psychoplastogen as a term. And that's why I consistently refer to metaplasticity, and I talk about open state engram modification as the mechanism rather than focusing on plasticity, because it's sort of it's a tricky concept to explain to people the difference between the two. But I do want to be clearly differentiated from that. So let me just double check the time. Okay, we're good. So then I think probably, if you're like me, you got interested in psychedelics when you realized that the LSD molecule looks just like the serotonin molecule, and that the two things might be working, you know, the LSD might be binding to serotonin's binding pocket. And actually, just for some historical funsies, you know, the serotonin 2A receptor was actually discovered because it bound to radio-labeled LSD, and this was work done by our Johns Hopkins' very own Saul Snyder early on in the days. And basically, more recently, there has been this beautiful paper by Brian Roth's group showing the crystal structure of LSD bound to the serotonin 2A receptor. And, you know, I think, you know, it got me really excited. Because it sort of explained why LSD has these very long lasting effects, because when it sits in that receptor, it gets sort of locked in. And when it does that, it triggers, probably, it looks like it's triggering beta-arrestin instead of regular G protein coupled signaling. And so it's thought that that beta-arrestin signaling might be responsible for all of the psychedelic effects of LSD compared to, for example, when serotonin binds to its receptor, right? And so that's exciting mechanistically, but, you know, if we try and look at how to classify psychedelics based on binding targets, some people have tried to sort of make a thing about, you know, serotonin psychedelics versus non-serotonin psychedelics. And I'm not really convinced that that's, you know, mechanistically going to lead us to the right answer, but because there are other psychedelics that are not 2A receptor binding, nevertheless, they induce that shared altered state of consciousness. So, you know, obviously ketamine binds to NMDA receptors. IBOGANE binds to the kappa opioid receptor, but it also has affinity for the serotonin transporter. Psilocybin also has some affinity for the serotonin transporter. And then obviously MDMA's primary actions are for the serotonin transporter. So we wanted to take a look at this and look at the serotonin 2A receptor dependence of this critical period reopening. And what we found is that exactly as the Brian Roth paper would predict, you know, the serotonin 2A antagonist ketanserin blocks LSD-mediated critical period reopening. Interestingly, also blocks the psilocybin-mediated reopening, but this mechanism is not universal because MDMA induced critical period reopening persists in the presence of the serotonin 2A antagonist, as this ketamine-induced reopening, right? So ketamine and MDMA don't care about the 2A receptor, and this reopening is independent of that 2A mechanism. Similarly, beta-arrestin, so now we took the beta-arrestin knockout mice, and I'm not showing you any of the controls we did to show that they're normal in terms of social reward learning in general. But when we gave LSD in the wild type, we were able to reopen, but in the beta-arrestin knockout mice, we no longer saw any reopening. So beta-arrestin, again, the Brian Roth paper did not mislead us. The beta-arrestin 2 is required for LSD-mediated critical period reopening. Interestingly, also MDMA-induced critical period reopening requires beta-arrestin. But again, neither ketamine nor ibogaine requires beta-arrestin. So what that's telling us is that the receptor and the next step downstream biochemical mechanism is not the universal mechanism, okay? So this is sort of a busy cartoon, but I'll just run you through it. This is just ibogaine binding to kappa opioid. MDMA, I didn't put its receptor in. It's basically acting mostly through the serotonin transporter, LSD through the 2A, ketamine through the NMDA receptor, and then this serotonin 2A mediated beta-arrestin or other modes of triggering beta-arrestin signaling are required for MDMA and LSD, but not the other ones. And so we thought, well, okay, if it's not at the receptor level and if it's not at the immediate biochemical step below the receptor at this second messenger signaling, maybe it's something even more downstream than that, that's common to all of them, that can account for this shared ability to reopen the critical period. And given the sort of long-lasting durability of these effects, we thought, well, maybe something's happening at the level of transcriptional regulation. So what we did is we did RNA sequencing, bulk sequencing. We took samples from the nucleus accumbens after giving psychedelics, 48 hours versus 2 weeks after psychedelics, or non-psychedelics. So we also had the cocaine control in here. And this is a busy slide, but what I want you to sort of focus on is that when we divided up the genes that were, the expression levels of the different genes that we found, and we categorized them based on whether the manipulation would have put the animal in the closed state versus the open state of the critical period, we saw this pattern of 65 genes that were differentially expressed in the open state versus the closed state. Some of the genes went up, some of the genes went down. But what was interesting is that roughly 20% of the genes in this differentially expressed analysis were members, either direct components of the extracellular matrix or regulators of the extracellular matrix, okay? And so what we think is and by the way, if we did this same analysis, but we didn't control for cocaine, so we just said drug versus non-drug, then we didn't get any significant ontologies, and we had very little overlap between this data set and the other. So it's, again, the cocaine control ends up being super important for being able to disentangle what psychedelics are actually doing in a brain region like the nucleus accumbens, which also reacts strongly to cocaine. Okay, and so I didn't, go through all of the different genes, but I'll just kind of give you our working model, what we're thinking now, and this is sort of the future direction slide, what we're trying to test next. And so basically we have some evidence now that maybe what's happening is that probably through the activation of immediate early genes, we get increased activation of MMP16, which is a matrix metalloprotease that has a cleavage site for fibronectin. It chews that up, starts to dissolve the extracellular matrix, making that plasticity available again. And that after some amount of time, the cell tries to restabilize itself by producing more fibronectin and also producing more of this TRPV4, which we think is important for stabilizing the synapse and inducing more fibronectin. So we are on the hunt for that. We've got the knockout mice and the drugs and everything, and we're on the hunt to kind of show that. But that's where we're headed next. And I'll just remind you that that regulation of extracellular matrix was one of the things that people had suggested might be an important last step mechanism for a critical period closure for the ocular dominance plasticity critical period. And this one is super interesting, I think, for, Lauren is interested in this topic specifically, I think. So there was this paper that just came out, well, I guess it's not just anymore, but about a year ago, showing that if you give repeated doses of ketamine, so like 6 of them back to back to back to back, then you can reopen the critical period for ocular dominance, and you do it in a manner that regulates the extracellular matrix. And so we think that this is exactly what we would predict based on our idea that what psychedelics are the master key for reopening critical periods. And if that's true, if it ends up being the case that they are the master key, then we should be able to reopen other critical periods, such as the critical period for recovery from stroke. And so that is sort of the overarching hypothesis of our Fathom project, which Lauren mentioned in the introduction. And we think this massively opens up the variety and types of diseases that psychedelics might be used to treat. And so with that, I'll just thank all of the people who did the work. If you guys are looking to hire anybody for faculty positions, Roman Nardu is the is the postdoc who was really the leader from beginning to end on this project. He's just amazing scientist and he's on the job market. So you guys should snap him up if you have any interest. And then Genevieve Stein O'Brien is the mathematician and the person who did all of the RNA sequencing for us. She's a postdoc in Loyal Goff's lab. She is also on the job market if you're looking for a computational geneticist. And then all these other people in my lab who also worked on this project. We got reagents from Rick Doblin at MAPS, the United Drug Program, Harold Gaynor and Misha Pletnikoff, and then we got funding from these agencies. So with that, I can stop and take some questions.
Great, thank you so much for leading us through that so clearly. Such exciting work. So we have some questions coming in, but first, Dr. Yehuda, I wanted to ask something.
Oh, thank you, Lauren. I don't have to go first, but Dr. Dolan, that was just fabulous. That was just a really fabulous lecture. But I have two questions for you. One is that I'm not quite clear on the distinction between metaplasticity and the psychogen, the psychoplasticity concept, and what you don't like about it. I completely understand that to the extent that the information comes from kind of neurons in a dish and the genes you're finding are in the extracellular space, I get that completely. But it seems to me that one could make a parallel critique of animal studies, especially using the analogy that you're missing the behavioral component, which you yourself said is the most critical part, the therapy. So I think both of these concepts explain why the therapy will take and why it is important to introduce the therapy. So could you review that a little bit?
Sure. Yeah, I'm glad you asked that because it is a little bit of a tricky subject and it matters how we interpret it. Okay. So the difference, I mean, I've been working on an analogy for this to try and explain it to people, and it's still rough, but the best I've been able to come up with is the difference between metaplasticity and hyperplasticity is that metaplasticity is, imagine a parking spot, okay, a parking lot, and it's huge, and when you're young, your brain is young, it's more like a grassy field. It doesn't have any lines drawn on it. You can just kind of park wherever you want and you can pack it in with hundreds of cars to go to the, hippie festival that you're going to go to. But as you get older and you start going to work and you need to be more efficient, your parking lot gets, lines drawn on there and there's a fire lane and there's, it's organized and maybe, there are fewer parking spots because it's a little bit more organized and a little bit more efficient. And that decrease in the number of parking spots is what I'm calling metaplasticity. Now, what hyperplasticity or learning or normal plasticity or memories are the actual cars, not the lines on the lot, but the cars themselves. So if you wanted to have a memory for, your trauma, it might be, 10 black and orange, monster trucks in your parking lot. That's the trauma memory. Okay, so that's the car is the memory. Well, hyperplasticity is let's say the hyperplasticity is related to cocaine. Okay, so what hyperplasticity is, 1,000, I love cocaine. Cocaine is awesome. Cocaine makes me feel good. Cars, let's call them, Corvettes or something, 20 of those packed into your parking lot. That's hyperplasticity. What psychedelics seem to be doing is not hyperplasticity, so not packing in more cars, but erasing those lines for the parking spot so that you can fit in a few more parts if you happen to be going to, say, the Oxytocin concert that night, right? And so the erasing of the lines is what I think, the way I'm sort of imagining what metaplasticity is. And why does that matter? That matters because clinically, if it's hyperplasticity, then the drug is the action, right? Like the drug is the thing, right? You give the drug, and if the therapeutic effects of the drug are only parking in, I'm not depressed anymore cars, let's say yellow beetle bugs, right? If that's what the therapy is, then all you need to do to make somebody not depressed is pack the parking lot with yellow beetle bugs. But what I'm saying is, I don't think that that's what the psychedelics effect is. We don't have any evidence that there are more yellow beetle bugs in the parking lot. Instead, our evidence suggests that there are more parking spots. And if we give the patient the right therapeutic context, if we allow them to, bring up those traumatic memories, then they can rearrange the cars and they can move some of the monster trucks out, and there's more room available for, let's say, I don't know, I'm making up car types here. I have a Mini Cooper, I love it. Let's put a Mini Cooper in there, okay? So red Mini Cooper cars for social bonding and being able to enjoy social interactions again. And that matters because what we think is one of the other sort of things that I didn't mention is that when I was a graduate student, people would say, oh yeah, the goal is to come up with a drug that can be the master key. And I never believed in this master key critical period idea. I used to make fun of Hench for this, and I, sort of owe an apology because my argument was, listen, anything that could do that to the brain would either melt the brain, so disrupt the structural integrity of the brain, or it would be amnestic, or it would cause massive seizure, right? Based on our mechanistic understanding of critical periods. But what the psychedelics seem to be doing is circumventing that melting the brain problem because their effects are context dependent, okay? And so it seems to be that because of this metaplasticity idea, the only memories that get changed are the ones that are recently activated, not sort of a widespread erasure or a widespread crowding in with the other things. And at the level of developing new drugs for therapies, if your readout for whether you have a successful new drug is hyperplasticity, then you're going to engineer your drugs to look more and more and more like cocaine, because that's the maximum hyperplasticity effect, right? So I just think that using hyperplasticity as a readout is going to, and I'll be honest, we see just a tiny bit of hyperplasticity with ketamine, right? It's not statistically significant, but there's a trend there, which kind of goes along with, there's some abuse liability issues cropping up around ketamine as well, right? And so you certainly don't want to make ketamine more plastic or more hyperplastic, because you're going to make it even more like cocaine if you do that. That's our argument.
Thank you.
I'm not sure if my car analogy works. I'm still working on it.
I understand the argument. I'm still a little stuck on how you can make the claim about depending on what kind of memory it is from animal studies, but I will work on that. I think that.
Okay. Oh, so let me just go back and explain that data a little bit. So basically what we did is we're not modeling the therapeutic setting in the mouse, right? We're not trying to say that social interaction is the same as a therapist sitting there. We're not modeling PTSD in our animal model. We're not inducing a trauma and then trying to unlearn that traumatic response, right? What we are saying is that just like the therapeutic effects are context dependent, MDMA's ability to reopen the social critical period is dependent on the social context, right? And so that context dependence is the thing that we're able to use as a proxy for, the right context that you have to have for the therapeutic effects. Now, in terms of how we might imagine that scales across different critical periods is that if we're looking at the stroke critical period, for example, we don't think that the social context will matter at all, right? In that case, we expect that our critical period reopening for, motor learning won't matter if you give it in the social context or in the isolation context. It'll work both ways. But what will matter is whether or not you give motor learning as the context, right? So if there's a running wheel, we would expect to reopen a motor context, right? And so we think that the sort of social thing is a little bit, it's how we got into it, but it's a little bit of a red herring. And we want to test this concept of the engram or the context or the memory that has to be co-activated with this open state using some optogenetic tools to be able to simulate specific memory engrams and show that only the engrams that are co-activated with this open state of the critical period are then available for modification. And we think that isn't an animal model of therapy. It's just provides an explanation for why it is that therapy is context dependent.
Yeah, I buy that. I like that explanation a lot. And I think it's going to be very challenging to apply this work, to try to understand why therapy is such an important ingredient of how psychedelics work. I love that you were able to really address with data the question of whether the duration of the experience matters and whether it matters to have an altered state of consciousness, which are the two questions I get asked the most, and are the basis for the development of drugs without the trip, which I completely don't understand, and wanting to make short-acting drugs. So I really loved everything I heard. I think that you're actually doing the most important work, translational work in psychedelics, and the field just owes you so much.
You. You want to be my reviewer?
I think I have been at some point or another.
Well, thank you. That's very, very nice to hear. Thank you.
Thank you. Next, we have Hope Cronman.
Hi there. Thank you for that talk. Fantastic. I had a quick question, sort of mechanistically. You talked about the involvement of several receptor systems and signaling pathways. And given that you're studying in the nucleus accumbens, is there any evidence that there's relevance of dopamine signaling to this process?
Yeah, so, originally when we discovered sort of social reward learning oxytocin, we looked at whether or not oxytocin plasticity, cares about dopamine D1 versus D2 and medium spiny neurons. It doesn't. And it just doesn't care. Like, it's the same in both cell types, right? And we think that that's something specific to, what social reward is like, right? So sort of in a really, really rough sort of way, one idea about what the reward encoding properties of dopamine are versus serotonin is that dopamine is kind of, you can sort of think of it as like immediate acute reward. So, I want this chocolate cake, I want to eat it now, and it's delicious now. Whereas serotonin is more like, I'm gonna not eat this chocolate cake because I'm trying to lose some weight, and so I'm delaying the gratification of being able to fit into my jeans, for the immediate gratification of the chocolate cake. And if you think about, the work of social interactions, for the most part, it's sort of, I'm gonna help my neighbor, and then when I need salt someday, I can walk over there, right? That's just, I'm just really anthropomorphizing here, but that's sort of, the way I'm thinking about the nature of social interaction. But that doesn't mean that there aren't certain circumstances where social interactions are that immediate reward, especially for sort of pair bonding, and which mice don't do, so we didn't study it. But so we have some ideas around where dopamine might be. That doesn't mean that there aren't some interactions of dopamine with the psychedelics in other contexts, but in a way, we're sort of relieved that the dopamine system doesn't seem to be super important for the psychedelic effects, because whenever dopamine gets involved, then abuse liability starts to creep into the mechanism. And I think that that's something that we are grateful for, right? That the psychedelics don't have much of an abuse liability, right? For the most part. So that's kind of my hand wavy answer. Oh, and I'll just say one other thing that it reminded me, though, is that even though we focused on the nucleus accumbens, we are in no way suggesting that this is the only place where these drugs are acting. We think it's, an engram that's distributed across the whole brain. It's just that because we have the biggest handle on social reward learning in the nucleus accumbens, that's why we focused on that.
Great, thank you. It's helpful.
Great. Next up, we have Kathleen Jagodnik.
Hi, Dr. Dolan. Thanks for your fascinating presentation. That was really great. In your 2015 publication, Bridging the Gap Between Patients and Models, you had noted that translational approaches in psychiatry usually fail, and you had suggested some approaches to remedy that problem, including some training programs that had been established at Johns Hopkins and from the Barer Foundation. Since that paper was published in 2015, have there been any further developments to address this challenge?
Oh gosh. So that was like a Ernst Strungman Forum report, and it was sort of like a bunch of clinicians and a bunch of basic scientists all thrown into a room to kind of come up with some problems, solutions. And then, as I was the most junior person at that whole meeting, they were like, well, why don't you write it up? And so honestly, it was not really like, I was there mostly from the autism point of view and, kind of bringing to bear my knowledge of, clinical trials in autism. So I can speak to that, but I can't give you any kind of update on some of the other suggestions that were in that paper, because those were like pieces that other people brought in from their own expertise. But from the perspective of autism, I can speak to the fact that, when the mGluR trials for autism, which the mGluR theory was my PhD work in Mark Baer's lab. And, people were really excited, 26 labs reproduced the mGluR phenotype or, correction of the Fragile X phenotype. And, the preclinical world was like, we got it. And then it failed in human clinical trials. And I think a lot of the people who were on the human side of things were like, well, that's just because a mouse isn't a human and, studying mice is basically useless. I mean, that was sort of the general reaction that we got. But the people who actually did the human clinical trial wrote a paper later where they suggested some alternative explanations. So the one that is relevant here is that people had talked about how, in the mouse studies, almost every single one of the mouse studies that was done was either in utero manipulations or very early in development, whereas all of the human clinical trials were done very late in development or in adulthood, right? And at the time that the first author wrote about this, we hadn't discovered the social reward learning critical period, but she suggested that there must be a critical period that's relevant to autism that's closed, and that would explain why the human trials didn't work. And so this is Liz Berry-Kravis, and I think she's actively pursuing that line of research to test in humans whether if they had intervened before the closure of the critical period that they're hypothesizing exists, then interventions would work. But what we're doing is basically saying, well, if that was the problem with that clinical trial, then what we should be doing is measuring not the outcome of learning, but the process of learning, because what we're saying that the mGluRs are doing is the antagonists are doing are bringing back the ability to learn. And we want to make sure that when we do that biochemical intervention, the social critical period is reopened so that those biochemical rebalancing has the chance to influence the relevant social memory learning process with an open critical period. So it's not like a direct answer to your question, but it's kind of the only part of it that I am aware of in terms of progress that's been made to bridge the translational gap.
Okay, thank you so much.
Unfortunately, we're just at the hour, but I feel like we could go several more. We're having so much fun here. So thank you. Thank you so much for coming. We'd love to continue this discussion, hopefully sometime soon.
Yeah.
So many questions. But yeah, thank you. And thank everyone for joining us today and for being a part of this. We appreciate it.
Thank you so much for the invitation. Please email me if you have any other follow-up questions, anybody.
Thank you.
Right.
Thank you.
Okay. Bye-bye.
Bye.